We are going to get started with our first in the pipeline series for the evening and talk about downstream targets. And so when we talk about downstream targets, what we're really discussing are all of those kind of effects that we see because of that lack of the dystrophin protein. So as we kind of discussed on Wednesday night with inflammation, that without the dystrophin protein, we can see inflammation. We can see the replacement of muscle tissue with fatty tissue and scar tissue. We can have calcium imbalance, cellular energy depletion, and all of these things can contribute to the disease progression that we see. And they don't just affect the skeletal muscle. We also see this impacting the cardiac, so the heart being a muscle too. So the strategies that we are going to talk about here in this session are going to focus on providing benefits by tackling kind of these downstream areas. So we're going to hear about some therapies that look at improving the function of the mitochondria, and then we're also going to hear about a therapy that's looking at providing some benefit to the heart. And I think it's really important that we, think about these therapies because although restoring dystrophin is very important and something that we look forward to. We know that exon skipping and even gene therapy aren't going to restore 100% of the dystrophin in the muscles. And so we still have these areas that are going to need to be addressed. We're going to have to take these therapies in combination with other therapies. So it's really important to continue to see the development of these different types of potential therapeutics. So I'm going to be joined tonight to discuss these by Dr. Gerard Marek from Astellas Pharma, Dr. Shabir Hasham from Santhera Pharmaceuticals, Dr. Larry Markham, he's an MD and he's representing Cumberland Pharmaceuticals, and Dr. Ransi Somaratne from Epirium Bio. And so, as a reminder, if you have any questions for any of our panelists, please submit them in the comment box below and just indicate what sponsor you are addressing that question to. So Dr. Marek, I think we are going to get started with you.
Okay, thank you very much. I'm Gerard Marek from Astellas Pharma, and if we could go to my next slide, then I'm going to be talking about ASP0367, which was formerly known as MA0211. And this drug, the target that it's acting on is a protein called PPAR delta modulator inside muscle cells. And we potential benefit that 0367 may act inside the muscle cells to actually increase the mitochondria's ability to produce energy. And mitochondria is sort of the powerhouse inside each cell. We're talking about we've completed phase 1 studies in healthy volunteers, and this next upcoming study is a phase 1B study that will be opening soon. We hope to have it opening up actually approximately in November for screening. We'll be looking for boys ages 8 to 16 to volunteer for this study. Some of the endpoints we'll be looking at is a novel one called a 6-minute cycling test, which is simply is an arm cycling test. Some of the secondary measurements include the pull, 2-minute walk test, respiratory function, and accelerometer, as well as other biomarkers. And we've been assigned to orphan drug designation from the FDA. And if the safety and preliminary efficacy signal in the upcoming phase 1B do work out, then we will be investigating efficacy and safety in phase 2 and phase 3 trials in consultation with the FDA. Next slide, please. So, as I mentioned, A0367 is being investigated to determine whether it can turn on PPAR delta pathway. And this PPAR delta pathway regulates the mitochondria on different genes in the cell. And when this pathway is on, the mitochondria are able to use fatty acids more efficiently, and more mitochondria are made. The mitochondria are working better, more efficiently. So this use of more fatty acids for energy results in increased energy production inside the cell. Next slide, please. So this phase 1B study is going to be a randomized, double-blind, and also placebo-controlled. The primary purpose is really to evaluate the safety and tolerability of ASP0367. We'll also be looking at how the body processes the drug, as well as I mentioned, how the drug affects function, looking at this arm cycling test. And trial sites we plan on opening in November of this year. And to find a participating site near you, you can visit the clinicaltrials.gov and search NCT 041-848-882. Or you can contact Astellas Pharma at the number that provided here, 800-888-7704. Next slide, please. So as I mentioned, for the study schedule, we have a 4-week screening period. This is followed by 2 weeks on a low dose of the drug, and then 10 weeks on a higher dose of the drug. And so this happens both for those subjects on active ASP0367, and about half as many subjects will also be on placebo during this 12-week period. After this double-blind placebo-controlled part, all subjects can go on to receive active ASP0367 for additional 12 weeks, and there'll be a 4-week follow-up. We have approximately 15 visits throughout this study, and what we just put in an amendment to our protocol, which has made a change where we've we've made about a little bit over half of these visits into home visits, just to minimize having to go into the clinical site, just as has been true for many of you in terms of normal clinical care as well. Next slide, please. So who can participate in this study? It will be boys 16 years old, having obviously a diagnosis of Duchenne muscular dystrophy, also taking stable doses of corticosteroids and any cardiac therapy that you're on, and having some trouble walking quickly as defined by the 10-meter walk test during your first screening visit. But you need to be able to move your arms up at least to the shoulder height. Exclusion criteria include acute illness or serious infection or other problems suggesting you're not healthy enough to participate in the study. Also having heart problems, like your heart not pumping well, where the clinical term is an ejection fraction, not being good enough, or long QT interval, meaning the heart doesn't send electrical signals well enough, or having high levels of cardiac troponin on a lab test, suggesting there may be other issues with heart cells. And also exclusion if you use certain types of drugs within the last 4 weeks of the study, or having kidney or liver problems, or currently receiving an investigational drug, or if you've ever taken an exon skipping or gene therapy drug. Next slide, please. So then what happens at the study visits? We'll be looking at your general health by Dr. Checking at study visits and blood and urine tests. We'll be looking at how your heart is doing with ECG echocardiogram, doing breathing tests, pulmonary function tests, how well you move your body, such as in the arm cycling test, performance of the upper limb test, 2-minute walk test, and other tests at home, including video tests and activity monitor. We'll also be looking at how your body uses the drug in terms of blood and urine tests, and then how you feel and think, a range of tests, including surveys, diaries, and interviews, and also looking at, like, for example, digit muscle test and other things. So I'd like to thank you for listening to our presentation, and I'd like to thank PPMD for the ability to present our study as well. Thank you very much.
Thank you, Dr. Marek. That was wonderful. One follow-up question I had for you. Around that exclusion criteria, I saw that was mentioned mental health problems or behavioral problems, and certainly, as you understand, because of the absence of dystrophin in the brain, we certainly have a number of patients impacted by psychosocial issues. diagnosed with different conditions. Can you define what is meant by mental health problems or behavioral problems for that exclusion criteria?
Well, that's really one where we're leaving that to the discretion of the PI, the investigator. So I mean, they just have to make the judgment whether or not the boy can actually engage with the staff and participate in the study and follow directions. So basically, it's can they follow directions for the study procedures. That's what it really implies.
Okay. Oh, thank you so much for that clarification. Wonderful. Okay, so we're going to move on to our next speaker. Dr. Shabir Hasham from Santhera is on with us, who's going to talk about what's happening with Santhera and their trials. And Dr. Hasham, are you there?
Thank you very much. My name is Shabir Hasham, Santhera Pharmaceuticals, and I'm delighted to present to you an update of our late-stage clinical development product, idebenone. And I just wanted to thank everyone for the opportunity to share the information with you. If I could have the next slide, please. So Santhera has been focused on investigating the advanced phases of Duchenne. Predominantly, we have an emphasis on trying to demonstrate the benefits of preserving lung function and the long-term benefits of doing so. We also have a product that targets mitochondria, and in our case, the product encourages electron shuttling across the respiratory chain, reduces oxidative stress, and increases ATP, and by doing such, allows respiratory muscle cells to function better and in some ways helps to prevent further muscle death. We have a late-stage development program. Data from our phase 3 Delos study was published in 2015, and this was in a population 10 to 18 years old. And these patients were in respiratory function decline, as we defined as having respiratory function of less than 80% predicted. Treatment with idebenone showed that we were able to statistically reduce the loss of respiratory function as measured by peak expiratory flow, but we also showed consistent benefit across forced vital capacity and forced expiratory volume. Importantly, we were also able to show by maintaining lung function and reducing the loss where possible, we actually reduced the risk of having infections, bronchopulmonary infections, and also the risk of hospitalization due to respiratory complications. Next slide, please. Whilst this is an important first step in building the knowledge about the potential long-term benefits of maintaining lung function, we've also been working quite extensively with academic institutions and doing modeling in natural history to try to understand what benefits could be brought in terms of long-term delays to clinically relevant milestones. I'm going to use this graph just as an illustrative example to try to bring across our key learnings. We examined natural history data in two populations, one in which respiratory function was declining faster, here shown in red, and compared it to one in which respiratory function was declining slower, here shown in blue. And what we were able to show was that those who were declining faster crossed clinically relevant thresholds of lung function that are known to predict an increased risk of infections and also the need for non-invasive invasive ventilation. What we were able to do in the modeling was to show by slowing down the rate of lung function loss, that population also crossed the clinically relevant thresholds, but later, and led to a delay in the need to start assisted ventilation. And this forms the basis of our continued work going forward in terms of trying to build the evidence base about a long-term benefit in preserving lung function. Next slide, please. We recently announced the completion of recruitment of our large phase 3 Sidros study in planned 266 patients. These were in patients 10 to 18 years of age, again, in respiratory function decline, having an FVC of less than 80% predicted, as opposed to the Delos study, which was investigated in patients who were not taking steroids. This study is looking at the combination of idebenone with glucocorticoids. We had an opportunity as part of the protocol to do a sample size re-estimation, and this really allows us to make sure that we have a sufficient number of participants within the study. The study was originally designed to have a 90% power, and that's the ability to detect a true treatment effect of the drug. When we did the protocol sample size re-estimation, we were able to look at blinded data. And what we saw that the respiratory function test, the FVC, the spirometry, and the peak expiratory flow tests performed much better than we had originally assumed in the study design. And so we went from a power of 90 to more than 99%, meaning that the study is very well powered to detect a true treatment difference. What we also were able to do was to determine that almost half of patients who were currently in the Sidros study had completed the 18-month observation period. And therefore, we're in a position now to conclude that we have enough data to conduct an interim analysis. Now, what that means is that if we are able to show compelling efficacy in this interim analysis, we're able to actually stop the study almost a year, if not a little bit more than a year ahead of schedule, and then start to have negotiations with the FDA for an NDA application somewhere in Q2 in 2021. Next slide, please. I just wanted to share some insights in the impact of COVID, and I'm sure many of my colleagues here have also been having to think about ways that we can manage COVID and the risk it has to the study. The team at Santhera, in collaboration with patient groups, but also with the community on a whole, have done a tremendous job in trying to participate in clinical trials. We were able to have strategies to deliver drug to home, remote monitoring, and also the possibility to try to extend visit windows. And I'm pleased to say that the impact of COVID on the Sidros study has been quite manageable, if not minimal. Thank you. Next slide. So in summary, Santhera continues to expand the data and knowledge that we have in terms of preserving lung function, delaying the loss over time, and the benefit that may bring in the long term in terms of delay to needing assisted ventilation. Data from the pivotal phase 3 Delos study had previously demonstrated that Puldysa has been able to slow respiratory function decline, reduce the risk of infections and hospitalizations, and we hope that the phase 3 Sidros study will continue to build on that knowledge base in a much larger group, but importantly in combination with glucocorticoids. Analysis done during the sample size re-estimation has given us the opportunity to stop this study early if we're able to show compelling efficacy in the interim analysis. And we hope that we'll be able to communicate to the community the outcomes of that by Q4 this year, which means that we may potentially be able to accelerate time to market by more than a year. And I'd like to thank everybody who's continued to participate in the study. I know it's been a very, very difficult time. And we just want to say thank you on behalf of the company and investigators and sites for the continued commitment that will help potentially to bring this study to an early closure with an outcome that could bring a therapy to market a year in advance. With that, it's just to say thank you to PPMD for the opportunity to share this information with you. Thank you, Ryan.
Thank you, Shabir. I really, really appreciate that presentation. That was really helpful. For some in the community who may not have been following for many years the work you all have been doing, could you sort of talk through your thoughts about why, with the original phase 3 meeting an endpoint, which was, I believe, the first trial to do that in Duchenne, why the FDA may have been reluctant to move forward with that, and explain to them sort of what happened after that and the path you took?
Sure. Thank you for the question. It's a very relevant one. So with Delos, we built on the experience of the previous phase 2 Delphi study in which we had been able to show a treatment effect originally in peak expiratory flow. And we used that as the basis to design the phase 3 study. And at the time, of course, FVC as measure was the preferred measure for the FDA. But in discussions with the FDA, we decided to proceed with the Delos study. But since then, we've been trying to establish the evidence base to show that actually PEF as a measure, clinical measure, is interchangeable with FVC. And that's the current focus, certainly within Europe and the regulatory strategy and submission there. We had a very clear understanding from the FDA that Sidros was the study that they would be interested in. And that's why we're so excited that we have the potential now to bring it to an early termination. Of course, it uses FVC, which is the preferred measure for the FDA, as well as using peak expiratory flow. So both studies are actually supportive of each other. And so we will have 2 studies to be able to show in a phase 3, obviously randomized control study, the benefit of Puldysa in terms of trying to delay respiratory function loss.
Thank you. That was extremely helpful. And I just want to acknowledge you all have done a lot of work to advance drug development for the non-ambulatory population. And we truly appreciate that. And we know that done a lot around endpoints that are needed to be advanced for this population. So we are grateful for that. Okay, so we are going to move on to our next speaker from Cumberland Pharmaceuticals, Dr. Larry Markham. Are you there? And please take it away.
Yes, I am here. So I'm Larry Markham. I'm a pediatric cardiologist living in Indianapolis at Riley Hospital for Children. And I've been working with Cumberland for the last couple of years. So next slide. I have no direct financial conflicts. Next slide. So we've been working on looking at ifetroban in the Duchenne individuals. So ifetroban blocks the thromboxane prostanoid receptor, which is expressed on many muscle cells, but also platelets and immune cells, and contributes to fibrosis in the heart and lungs. And so treatment with ifetroban has the potential to prevent fibrosis and inflammation. It's an oral medication, which is administered once daily, and safety is well established in other disease indications. Next slide. If you would like some more background on the basic science information, you can check out poster 14, which has been uploaded. And this is work that I and a team collaborated on with investigators at Vanderbilt. But this is a cartoon of the pathway. And you can see there at the top, aspirin is one drug that also interacts in this pathway. But ifetroban blocks all of the cell surface receptors, and as such, blocks some of the pro-fibrotic signaling pathways. Next slide. So working with PPMD, Cumberland, Fight DMD, colleagues at Vanderbilt, this has been a process that has been going on just over 3 years. And last fall, this was awarded the first DMD trial, awarded an FDA orphan grant to study this drug in Duchenne, and was also sponsored by Fight DMD, which is a foundation based in Nashville. Next slide. This is probably the most important slide, is the study design. So this is a phase 2 with the objective to evaluate safety, efficacy, and pharmacokinetics. 2/3 of the subjects will be randomized to ifetroban, 1/3 will receive placebo. The treatment duration will be 12 months, and there will be 3 visits. At visit 1, will be randomized individuals to low dose or high dose. Some of the criticisms centered around, if you take individuals who all have normal heart function, how will you tell if it works? And so we have 2 groups of individuals getting treated, those with mild cardiac involvement or no cardiac involvement, and those who have a little bit more moderate cardiac involvement, to see if we can rescue those who already have significant cardiac impairment. Next slide. There are some key eligibility and study features. This is an option for all Duchenne boys greater than 7 who can tolerate a cardiac MRI, regardless of mutation type, ambulatory status, or steroid use. There is no upper age limit, and even those who are on exon skipping therapies will have possibility. And because it includes patients with all ambulatory categories, there is no walk test and no biopsy. Next slide. This is a map of the sites. Currently, Children's National in DC is up and running. As Shabir said, we've all been impacted by COVID and how we can get things going. Here at Riley, we're close. We're probably the second site that will be up in this list, and all we need to have is our site visit. And then there are several others that are coming on rather quickly now that we have returned to some research activities. If you do have an interest, there is the address and email for consideration of being involved in the study. And again, as everyone says, I know the conference has been significantly impacted. And so we just appreciate the opportunity to be involved with the community in this way. And I think the next slide is just questions and thank you. So you can click through those. Thank you.
Great. Thank you, Larry. Really good to see you virtually. Just one quick follow-up question before we move to the next speaker. I think families are probably wondering, are you envisioning this is going to be in combination with standard of care cardiac medications in the future, and that's sort of the plan for treatment?
Yeah, I think as we've looked at this and looked at things over time, I think we are starting to understand that scar tissue or fibrosis in the heart is a very early phenomenon, and a lot of the treatments that we do, ACE inhibition, beta blockers, and eplerenone or spironolactone, they all target fibrosis, but in different ways. And so I think if we have another option to add to that collection of drugs, then I think it will be a good thing.
I definitely agree. I certainly agree. Combinations are needed, and we appreciate that. Okay, we will speak to you on the panel in just a bit. Our last speaker of this session is from Epirium Bio. Ransi, are you there? Wonderful.
Yes, I am.
Please take it.
Can you hear me? Great. Thanks very much. First slide, please. Thanks very much. I'm Ransi Somaratne, Chief Medical Officer at Epirium Bio. As Eric so nicely outlined at the top of the presentation today, the lack of dystrophin leads to a number of downstream consequences that end up leading to fibrosis, fat deposition, and we see this as a vicious cycle. And the hypercontractility-induced injury in the skeletal muscle fibers leads to inflammation, loss of mitochondria, loss of mitochondrial efficiency, accumulation of reactive oxygen species, fibrosis, which leads to more damage, and this cycle continues. Next slide, please. So what we've learned, and our lead candidate is called EPM01. We believe that this compound shows potential benefit for patients with both Duchenne and Becker muscular dystrophies. And what we've learned through a couple of small pilot studies using EPM01 and an earlier, less potent compound is that there appear to be multiple potential benefits for these patients, such as improved energy balance in cells through enhanced production of mitochondria and enhanced mitochondrial function, a reduced inflammatory response in muscle, a reduced amount of oxidation injury, improved balance in the proteins regulating muscle regeneration and growth, including follistatin and myostatin. We've noted the increased production of nitric oxide. In the Becker study, we noted increased expression of key proteins such as dystrophin. We've seen encouraging data on cardiac function, including effects on brain natriuretic peptide and troponins, and we've also seen encouraging trends in exercise capacity. Next slide, please. So through the multitude of effects that we've noted, we believe that the EPM01 and this class of medication could improve all the various stages of the cascade of events occurring after loss of dystrophin and potentially lead to clinical benefits in patients. Next slide, please. So where are we currently? We have just started up a small study in patients with Becker muscular dystrophy. We're looking to enroll 20 patients between the ages of 16 up to the age of 60. We're primarily looking at the safety and tolerability of 3 doses in anticipation of the future trial. We're going to be following these patients up for a total of 1 year on drug. We'll be doing things like the timed muscle function test, which you're all accustomed to in this type of clinical trials, looking at key muscle proteins and other biomarkers. We'll be looking at cardiac assessments, including cardiac MRI via a core lab, and BNP. And we're really now moving the program towards studies in Duchenne muscular dystrophy. We're still trying to work out the clinical research phase, the target ages in Duchenne, and the endpoints that might be of importance in our studies. We're also working with regulatory agencies in defining the pathways for both the Becker and Duchenne programs. I should note that we've been granted orphan designation for Becker and Duchenne muscular dystrophy by the FDA. Next slide, please. So key takeaways, we believe that our compounds have a unique mechanism of action that's directly relevant to Duchenne muscular dystrophy. It seems to restore bioenergetics and muscle structure. We've now, through a variety of prior studies, have safety in over 100 patients, both adult and pediatric, across a multiple set of diseases. We have been encouraged by the types of biomarker movements we've seen in both Duchenne and Becker muscular dystrophy. We've seen encouraging signals of efficacy in the peripheral and cardiac muscle of Becker and Duchenne patients. We're currently in a phase 1 dose escalation study in biomarker and cardiac function assessments in Becker, and we're driving now towards studies in Duchenne patients. I'd like to thank all of you for your attention and thank PPMD for this opportunity to speak.
Wonderful. Thank you, Ransi. That was a really great presentation. So this community has supported some of this early research years back, and I just wanted you to explain a bit that the term they've been used to for the years is epicatechin. So it's something that wasn't said in your presentation, but the community actually funded this early work through PPMD. So I wanted to acknowledge that, but also just have you explain a bit. Was it reformulated over time, or from the original work, or tell us how that path or why it took to now to get to this stage? I think it'd be important for them to hear.
So, and we're grateful for the partnership with PPMD over the years. So our company used to be called Cardero Therapeutics, and Cardero has been around for about 10 years, and our co-founders worked very hard, and it was through the support of organizations like PPMD that we've now gotten to where we are. We rebranded the company, and we had a very successful Series A financing round in December of 2019. And yes, the core compound is epicatechin. We believe that these two stereoisomers of epicatechin minus and plus epicatechin have unique physiologic properties, as I've outlined so far. And both of these, the minus and the plus, have been studied in pilot studies before. However, we've come to realize that the plus isomer is much more biologically active and has physiologic properties that the minus isomer doesn't seem to be able to replicate, at least with the types of doses that you'd want to do in patients. So the company's been working hard on a way to refine and manufacture the plus isomer, which is extremely difficult to find in nature. And so EPM01 is the plus isomer, which appears to be significantly more potent and significantly more biologically active. And so that's the reason why we're taking that compound into clinical development.
Thank you, Ransi. That was extremely helpful. And, we like more potent and more biologically active. I think those are two really good things. All right, we're going to now kick this to Eric, who's going to do the Q&A of the panel. Eric?
Yeah, so just a reminder, if you have questions, please submit them to the box below and indicate which sponsor you're directing this towards. And this is one that I'm going to kind of pass around to the whole panel here. One, again, we love to see that you're all testing your products and investigating them in some of these populations that don't always have the opportunity to participate in clinical trials. But there have been some questions as well about whether these products could benefit use at a younger age, and if there's any plans to investigate these in a younger population of Duchenne. And in the case of Epirium, maybe if you have a timeline for when you may get to Duchenne. So Dr. Marek, I will start over with you.
So Yes, initially we're looking at ages 8 through 16 and looking at sort of the late ambulatory and non-ambulatory phase. And that's largely a strategy just so that we can try to, as rapidly as possible, conduct an understanding of safety and efficacy, given the fact that so many boys are enrolled in other types of studies early on. So it's for that reason. But down the road, we'll be wanting to look at the entire range, yes.
Great, thank you. Dr. Hisham? Oh, I think you might be muted.
Sorry.
No problem.
Sorry. Sorry, Eric. Yeah, no, I agree with Dr. Marek. Although the majority of our data is in the older population, there is no scientific reason why we wouldn't have the same or similar benefit in a younger population. Of course, it needs to be proven. We have some initial data from our phase 2 study, but certainly it's something we would look at in the near future.
Great, thank you. And I know, Dr. Markham, you indicated there's a challenge in kind of detecting efficacy with the younger ages, but is this something you still think may be able to be beneficial treating at an earlier age?
Yeah, definitely. I mean, I think if we can see a signal of benefit in the 7, 8, 9, 10-year-old who can undergo an MRI, in my mind, there's nothing to preclude going even younger. And at that point, I think you're just talking about treatment. So I don't even know if it is necessary to study it in a 6-year-old who can't do an MRI if the signal is valid.
Great, thank you. And Ransi, if you could maybe give an estimate of when you think you might be able to look at the Duchenne population.
Well, as I said, we're working towards this now. In fact, we're looking at 2 or 3 different options. I can't make any promises, but I would love to be able to say that we would be in the Duchenne population within a year. That's certainly by this time next year. I think that's a goal that I would set for ourselves.
That's great. And again, I know there's a lot of appreciation too from our Becker population who really don't always get the same opportunities, and so I know they're all grateful for that. Mm-hmm. Dr. Marek, so we've been talking a lot about COVID, and there was a question. Could you maybe explain a little bit more about what some of those, that you talked about the at-home video and some of the remote monitoring that's being developed for the trial. Could you maybe explain what is involved in that?
Sure, I'd be more than happy to. For some of the remote monitoring, what we've done is like the things that we'd be doing otherwise, safety visits to the clinic, we've moved them to being conducted remotely by qualified nursing staff. And in terms of the video monitoring, that's sort of work that we're actually doing, looking at certain tests of how the patients are doing, where we're collaborating with Casimir on those efforts. So it will allow us maybe to bring some, I think certainly Casimir is looking at how they can bring new measures to patients. So I think everyone in the field is excited about those possibilities.
Yeah, we had a lunch and learn session that's going to be recorded and released next week for anyone interested in that work. So thank you for mentioning that. Go ahead.
Yeah, and if we just had a question come in for Epirium, although I do think we could ask this question to everyone on the panel as well. But for your study with Becker, is there any possibility about kind of expansion to female manifesting carriers?
At this point, no. We are enrolling patients with Becker and Becker-like mutations, but at this point, we don't have any plans to expand to female manifesting carriers, but I'd be happy to talk to somebody about it if there's a significant enough desire to do so.
Great, thank you. And I don't know if anyone else has any could comment on that, if their product may be possible to use in that population or any plans. Dr. Hisham?
It's certainly possible. Of course, our trials are coming to a conclusion, so we'd have to look at the post-approval setting for any potential to do that.
Okay, great, thank you. Dr. Markham, in your in the open label extension, can you share what dose you expect patients will be receiving during that phase?
So we initially had planned an interim analysis after the 6-month interval to help guide what that extension would be. And so if there is a dose that seems to be more efficacious, then that would be the dose that would be chosen.
Okay, great, thank you. We also have had a number of questions come in just about combination of therapies. And I think that's something that we talk about a lot. It's something that we think is important. And did notice that is part of kind of the current exclusion criteria in the Astellas trial that any of the exon skipping therapies, is part of that exclusion criteria. Could you maybe explain some of the rationale behind that decision, Dr. Marek?
Well, essentially, we are very early on. So for this initial phase 1b study, we're excluding that just because there may be some change during the study with the exon skipping therapies, and it's going to be very, very hard to be able to look at that extra variation in a very small study like this. Down the road, as we get into larger phase 2 and phase 3 studies, we'll be certainly reconsidering this strategy.
Okay, great. Thank you. And then actually, we had a follow-up question as well come in on that. Is there a plan for an open label extension in your trial?
Yes, after the first 12-week period, then all patients are allowed to go into a 12-week open label extension, yes. So also that will be available to all subjects. So the subjects that are originally on active drug will be able to get an entire 24-week period. But even the ones on placebo will have a 12-week potential exposure to the ASP 0367.
Okay, great, thank you. And this one came in for Epirium. So in your next study phase, are you planning on having just U.S. sites or looking towards a global pathway?
In the Becker or?
In the Becker trial.
Yeah, no, we would very much be looking to do a global study in our follow-on to this current trial.
Okay, great. Thank you. And so we actually are just about at time, so I'm going to say thank you to all of our panelists. We really appreciate you taking the time to be with us today. Anyone whose questions we weren't able to get to, again, we will pass those along to the sponsors. And so thank you all for joining us this afternoon.